Welcome to the fascinating world of cell signaling. Imagine you are living in a massive, bustling city where millions of people need to coordinate their daily tasks perfectly. If no one could communicate, the city would instantly fall into absolute chaos. Your body is exactly like this city, made up of trillions of individual cells that must constantly talk to one another to keep you alive and healthy. Cell signaling is the magical postal service of your body, allowing cells to send, receive, and interpret chemical messages. We're going to explore exactly how this beautiful system works, step by step, so you can fully master it. Now that we understand why communication is vital, let us look at how it actually happens. In molecular biology, cell signaling refers to the complex network of communication pathways that govern basic cellular activities. The process typically begins when a specific chemical messenger, known as a ligand, is released. This ligand travels until it encounters a target cell equipped with a specific receiving protein called a receptor. Upon binding the ligand, the receptor undergoes a conformational change, which is a fancy way of saying it changes its three-dimensional shape. This shape change triggers a cascade of intracellular events, ultimately leading to a specific cellular response. Understanding this is critical because when these pathways break down, it leads to severe diseases like cancer and diabetes. To truly understand this binding process, we must look at the mathematics behind it. This brings us to the dissociation constant, a crucial concept in receptor kinetics. The formula shows how receptors and ligands interact dynamically. In this equation, R represents the concentration of free, unbound receptors on the cell surface. L represents the concentration of the free ligand floating outside the cell. RL represents the concentration of the receptor-ligand complex, which is the active state where the message is being received. Finally, KD is the dissociation constant, which tells us how tightly the ligand binds to the receptor. If the value of KD decreases, it means the binding is very strong, whereas a higher KD means the ligand easily falls off the receptor. Building on the dissociation constant, we can now determine exactly how much of the cell's communication network is activated. This is done using the fractional occupancy formula. Here, the variable y represents the fractional occupancy, which is the percentage of total receptors that are currently bound to a ligand. The symbol L is, again, the ligand concentration, and KD is the dissociation constant we just discussed. This formula is incredibly powerful. As the value of L increases, the fraction y approaches the number 1, meaning 100% of the receptors are occupied. Conversely, if L drops to 0, the fractional occupancy also drops to 0, meaning the cell receives no signal at all. Now that we have our formula, let us see this with a concrete example. Let us imagine a scenario where a cell is exposed to a hormone ligand. We are given that the dissociation constant for this specific receptor is 20 nanomolar. The current concentration of the hormone ligand outside the cell is also 20 nanomolar. We need to calculate the fractional occupancy, Y. We substitute the given value of 20 for L into both the numerator and the denominator, and 20 for KD into the denominator. Adding the denominator gives us 40. Dividing 20 by 40 gives us a final value of 0.5. Physically, this means that exactly 50% of the receptors on the cell surface are currently bound to the hormone. Let us take this a step further with a real-world medical scenario. A pharmaceutical scientist wants to design a drug that activates exactly 80% of a specific cell receptor to treat a disease. The drug has a known dissociation constant of 15 nanomolar. we need to calculate the exact ligand concentration required. We start with our standard fractional occupancy equation, substituting 0.8 for Y and 15 for Kd. To solve this, we multiply both sides by the denominator, giving us 12 plus 0.8L equals L. Subtracting 0.8L from one full L leaves 0.2L. Dividing 12 by 0.2 gives us 60. The scientist must administer a drug concentration of 60 nanomolar. Having calculated these values, let us visualize this relationship on a graph. What you are looking at is the receptor-ligand binding curve. The horizontal axis represents the ligand concentration, while the vertical axis represents the fractional occupancy, Y. Notice how the curve starts at 0 and rises steeply as ligand concentration increases. However, it does not go up forever. It eventually levels off, approaching a maximum value of 1, represented by the dashed horizontal line. A critical point on this graph is where the fractional occupancy is exactly 0.5. If you trace this point down to the horizontal axis, it aligns perfectly with the dissociation constant, Kd. This visually proves our earlier rule. When ligand concentration equals Kd, half the receptors are occupied. Now that we understand basic receptor binding, let us look at a specific, highly important type of receptor pathway. Imagine a relay race where runners pass a baton. The G-protein coupled receptor, or GPCR, pathway works exactly like this cellular relay race. GPCRs cross the cell membrane exactly 7 times. When a hormone binds to the outside, the receptor activates an internal G protein by exchanging GDP for GTP. This activated G protein then stimulates an enzyme called adenylyl cyclase, which produces cyclic AMP. This cyclic AMP acts as a second messenger to flood the cell and activate protein kinase A. This pathway is fundamentally important because it controls almost every aspect of human physiology, including your fight or flight response to adrenaline. This relay system is not just about passing a message, it is about making it louder. This brings us to the signal amplification formula. The symbol A total represents the total number of final activated molecules produced from just one single ligand binding event. The variable N sub G represents the number of G proteins activated by one single GPCR. The variable NCAMP represents the number of cyclic AMP molecules produced by one activated adenylyl cyclase enzyme. Finally, NPKA represents the number of target proteins phosphorylated by one activated pKa enzyme. As any of these individual variables increases, the total amplification grows exponentially. This explains how a tiny amount of hormone can create a massive cellular response. Let us calculate the massive power of this cellular relay race with an example. Suppose a single molecule of adrenaline binds to a GPCR on a heart muscle cell. This single GPCR activates 100 G proteins. Each activated G protein stimulates an enzyme that produces 1,000 molecules of cyclic AMP. Finally, each cyclic AMP activates a pKa enzyme that phosphorylates 50 target proteins. We need to find the total number of phosphorylated target proteins. We substitute 100 for Ng, 1,000 for NCAMP, and 50 for NPKA. Multiplying these three numbers together gives us 5 million. Physically, this means a single molecule of adrenaline results in 5 million activated proteins, making your heart instantly pound harder and faster. To fully appreciate this massive amplification, let us look at a schematic diagram of the entire cascade. Here, you can see the cell membrane separating the outside environment from the inside. Notice the GPCR weaving through the membrane 7 times. The ligand, epinephrine, is bound to the top. Below the membrane, you can see the G protein complex splitting apart, with the active subunit moving over to bind with adenylyl cyclase. The diagram shows ATP being converted into cyclic AMP molecules, which then activate pKa. Finally, you can see pKa entering the nucleus at the bottom to activate the Krebs transcription factor on the DNA. This visualizes the masterpiece of biological engineering that turns a faint whisper into a roaring shout. We have seen how GPCRs amplify signals at the membrane, but now let us contrast this with a completely different type of signaling pathway. Imagine a VIP executive who bypasses the front desk and walks straight into the CEO's office. The nuclear receptor pathway works exactly like this. The signaling molecules here are special lipid-soluble hormones like estrogen and testosterone. Because they are hydrophobic, they slip right through the cell membrane without any assistance. Their receptors are located entirely inside the cell. Once bound, the activated nuclear receptor acts directly as a transcription factor, binding to specific DNA sequences to turn target genes on or off. This pathway is absolutely vital for long-term cellular changes like growth and development. Because these receptors directly control gene expression, we can model their effects mathematically. This is the transcription rate equation. The variable RTX represents the current rate of gene transcription, measured in messenger RNA transcripts produced per minute. The symbol Rmax represents the absolute maximum possible rate of transcription when all DNA binding sites are fully saturated. The variable CNR represents the concentration of the active ligand-bound nuclear receptor complexes inside the nucleus. Finally, Kact is the activation constant, representing the concentration required to reach exactly half of the maximum transcription rate. If the concentration of active receptors increases, the transcription rate will increase until it eventually plateaus at the maximum limit. Let us apply this transcription rate formula to a biological scenario. A cell biologist is studying the effects of testosterone on muscle cells. The maximum transcription rate for a specific muscle growth gene is 120 transcripts per minute. The activation constant is 30 micromolar. The current concentration of active nuclear receptors is 10 micromolar. We need to calculate the current rate of gene transcription. We multiply the maximum rate of 120 by the active receptor concentration of 10 in the numerator. In the denominator, we add 30 and 10 to get 40. 10 divided by 40 is 0.25. Multiplying 120 by 0.25 gives us 30. The cell is currently producing 30 transcripts per minute. We have covered a tremendous amount of ground today, so let us review the key takeaways. First, we learned that cell signaling begins with a ligand binding to a receptor, and we can predict this interaction using the fractional occupancy formula. Second, we explored the GPCR and cyclic AMP pathway, discovering how a single molecule can trigger a massive, rapid cellular response through exponential signal amplification. Finally, we examined the nuclear receptor pathway, where lipid-soluble hormones bypass the membrane to directly rewrite gene expression for long-term changes. Understanding these distinct communication pathways gives us the power to comprehend human biology and even cure devastating diseases. You now have a solid foundation in cell signaling.